Okay, good morning again, everybody, and welcome to the next talk. Dr. Hal Alper is the Les and Sherry Stewart Endowed Professor at Chemical Engineering and the Director of the Center for Biomedical Research at the University of Texas at Austin. He earned his PhD in chemical engineering from MIT and went on to postdoctoral positions at the Whitehead Institute and Shire Human Genetic Therapies. His current lab focuses on metabolic and cellular engineering in the context of biofuel, biochemical, and biopharmaceutical production in an array of model host organisms. He has won numerous prestigious prizes and is an elected fellow of both the American Institute for Medical and Biological Engineering and to the National Academy of Inventors. This morning, he'll be presenting on integrated cell and cell-free metabolic engineering approaches for rapid prototyping of genes and pathways. Take it away, Hal.
All right, thank you very much for that introduction, and thanks to obviously for all the organizers for this conference, especially Nigel, for the invitation to be able to talk a lot about our work that we have ongoing collaboratively with JGI. And title, as we just mentioned, is this integration of cell and cell-free metabolism. And really as a motivation for that, and kind of we heard a little bit of motivation before in that in the prior talks in terms of looking at increasing productivity of cellular production, we really see that there are two alternatives to increase this productivity. We can use a cell-based process, and this is the kind of bread and butter to some extent of the biotech industry. These cells are complex, self-propagating metabolic factories that can enable the production of fuels, chemicals, pharmaceuticals, proteins, just biomass in general. Right? The challenge, though, is that they have very long development timelines. It takes a while to engineer a cell. We can't very easily rapidly prototype these systems. On the flip side, if we actually took that cell, opened it up, spilled its guts into a tube, we have these cell-free based systems. And in these systems, we can actually do metabolism. And we can utilize the fact that these are self-contained catalytic factories that have the ability to be rapidly prototyped, meaning we can mix and match. We can do things in terms of the chemistry, in terms of additives that we wouldn't necessarily be able to do with a whole cell that needs to be able to maintain viability. However, how do we parse between these types of two avenues? How do we go from a cell into a cell-free system? How can we actually engineer that cell to make a better cell-free system? And in turn, how do we actually take and use the information from that cell-free system to be able to go back and redesign and improve in vivo cellular system? And really, this is this integrated approach for rapid prototyping that we've been working on developing over the past couple of years. So I'm going to start off really is two distinct stories. One, talking about how we can make significant advances in the rapid prototyping of in vivo-based systems through especially CRISPR-based approaches. And this took advantage of a guide RNA library that I'll talk about that was synthesized at JGI. And then how we can use these rewired cells to create rewired metabolic lysates that can be used to rapidly prototype pathways of interest. And this is work that is sponsored by the JGI project. So starting with the cell-based system. So CRISPR, needless to say, has been talked about in this overall conference so far, is a great tool to be able to endow newfound control of gene expression. We have ways to be able to either encode changes into the genome, to be able to change activation, to change repression in tunable, programmable, modular type of fashion. And really, this is enabling us to be able to begin to think about rapid prototyping of cell-based systems. And this is allowing us to go into organisms that we ordinarily would not have explored, and also into strain variation that ordinarily we would not have explored, mostly because of the very long timelines of development that most of these things take. So some of the work that we've done in terms of this, what we call stepping of the system, where we can actually place a DCAS9 activator or repressor linked together with a kind of targeting guide RNA to a promoter region. And for example, if we have a repressor that's very close to the start codon, we pretty much repress expression almost completely. If we begin to step that along further and further away from that start codon, we can actually begin to have less actuation of that repressive activity. So therefore an increase in the overall expression level until we hit about that basal level expression that that promoter would have been able to impart. And then if we switch that now to an activator and then begin to move that closer and closer to the start codon, we can actually begin to have tunable activation of that gene. So in the end of the day, we're able to get this large dynamic range of expression that's a programmable aspect by using this DCAS9 system. So really, this has enabled us to program and multiplex, meaning we can actually step along the way to any gene of interest in a programmable type of manner, change that degree of actuation, and then be able to measure that phenotypic response. So this allows us in an iterative fashion to debottleneck pathways. So we can test for our phenotype of interest, some metabolic overproduction, using this type of steps approach. Find that that bottleneck is because we can query this across all the different potential rate limiting steps within a cell or even a library basis that I'll talk about in a little bit, and then use that to develop our next strain. Once we've eliminated the bottleneck, we can retransform these already pre-made from the freezer, essentially, guide RNAs that are sitting around to iterate this process. And this allows us to go through iterative strain engineering in a very rapid fashion. So as an example for that, and a motivation for a project that we'll talk about a little bit later, is this idea of looking at the glycerol pathway in Saccharomyces in more aerobic conditions. So what we can do is kind of target some of these key genes of interest by using this STEPS approach, and we develop this guide RNA panel to a variety of these different genes, and then we can look at the overall response. So we can see here, as an example, with TPI, there's really no response with actuation, and there's beginning to be a response as we really strongly repress that gene. And that's kind of understood. If you knock out TPI, you're going to force half that flux from glucose to go down into the glycerol pathway. But we can start to scan all of the different targets of interest across the board, and we really see the one that really pops out is GPD1, where we find that stronger overexpression of that particular target is a essential part for increasing glycerol production. And indeed, that's really already been kind of shown as the major rate limiting step. In contrast, the next step, GPP1, actually is pretty much flat and doesn't have a response. However, once we now know our bottleneck, we can target that in and encode that into the cell, overexpress the GPD1, and then reintroduce this guide RNA library and find a different response pattern. Once we overexpress GPD1, we no longer see that repression of TPI1 is going to give any beneficial aspect of the flux, but yet we see that there is this linear response with respect to GPP1, showing that that's the next rate limiting step of the process. So what this allows us to do is go through iterative type of rounds, and we can achieve quite high overall titers, in this case, getting up to almost 30 grams per liter of glycerol from this strain by looking at these individual targets. So we started thinking about how can we expand this idea, right? So that was kind of our individual work where we looked at a specific pathway, designed these guide RNAs, went through these kind of targeted approach. So we wanted to kind of see if we can really synthesize a high information tiled steps library for yeast. And in doing so, we went to the stoichiometric model for yeast, found and designed guide RNAs for all of our major coding regions within this metabolic model, developed a script to be able to go through and find tiled guide RNAs that will allow us to have ranges of activation and repression for that. And then thanks to efforts and support from the JGI DNA synthesis program, we're able to create these guide RNA pools that we bind into roughly 4 different functional groups that really spanned a significant number of targets within yeast. So all of the 969 genes that were in this metabolic model. So what this enables us to do now is move from a targeted approach to an untargeted approach, or library-based selection. And in doing so, we can get these libraries where we can introduce these guide RNAs together with either an MXI or VPR repressor or activator, introduce those into cells, go through an enrichment step, whether it's growth enrichment, a product selection, or in work that will be forthcoming in the next couple of days in a new article on microdroplet-based selections, but really using this guide RNA library linked together with deep sequencing analysis, we can begin to see the enrichment of these various different guide RNAs, and we'll see points of optimality for the genotype and phenotype of interest. So what do I mean by that? Well, we enable this really bidirectional titration of gene expression. So we can do this in a pooled type of manner. So with these single guide RNAs, we can either have this range of downregulation or a range of upregulation. This allows us, when we introduce these libraries, to go through selections. So two selections I want to talk about today very briefly are one, growth on galactose, and two, growth on glycerol as carbon sources, just because these are easier growth-based selections. So when we do that, we actually can find the enrichment of guide RNAs and see that various different guide RNAs are clustered and kind of dominant for different types of levels of expression, meaning it's not always strongest overexpression or strongest knockdown or knockout, but rather there's intermediate optimal expression levels that really are important for our genotypes of interest. And just to really demonstrate that, if we look at the fold enrichments that we're getting from this desequencing analysis, we see that it's not just low expression, but sometimes intermediate expression that is important both for galactose and for glycerol. The important thing about this approach is that we're able to complement the traditional genetic approaches of strong overexpression and knockouts. So really, this allows us to uncover unreachable targets through this type of library-based approach. So if digging into that glycerol enrichment example as just a case study here, we were able to find targets for which the knockdown was much better than the knockout. So here's two examples for that, for TPS2 and IPK1. So two different targets that we had identified. The deletion in terms of growth is shown on here on the right. The growth that we have from the lower knockdown type of approach here in green compared to the blue for the control. And we see that the growth is better by far by knocking down a target rather than knocking out. We find also that intermediate overexpression is much better than strong overexpression. We get that here for GRS2 as an example. When we have intermediate overexpression, we have a much better growth curve than we have for very strong overexpression. So sometimes the optimum expression is not the highest that we can get, but something more intermediate to that. And this type of guide RNA tiling approach allows us to find that in a rapid type of fashion. And finally, the one that's interesting, kind of going along with the knockdown, is that sometimes the knockdown is functional, but the deletion is lethal. So, for example, with GPI18 rather, we found that when we knocked that out, we had no growth. It was a lethal mutation. This is an essential gene. But when we knock it down, we actually have a beneficial phenotype in terms of glycerol growth compared to that of the control vector. So really using these CRISPR-enabled libraries allows us to go through and survey a much broader span of metabolism and find key genes that relate to that improvement in flux. So we went through and we've been further developing the way in which we do this, the ease in which we do this, the kind of ability to use just a Pol2-based expression system, only a DCAS9 activator-based system, and really getting these large fold range of expressions, so really trying to streamline this overall process. And then also, as we move to this Pol2, we can actually look at having guide RNAs in a cassette, essentially, for yeast, so almost having an operon of guide RNAs. We've also done similar things by using T7-based expression on here. But really, this allows us to be able to multiplex these guide RNAs. And when we're able to do that, we can actually rapidly prototype cells. And we've been able to, for example, look at 5 different changes at the same time in yeast to be able to rewire this for the production of 2,3-butanediol, very rapidly being able to combine both knockdowns and overexpressions in that pathway. And when we do that, we can see that this design is portable. We can do this in SynPK, for example. We can throw these guide RNA array in there, and we get a very quick increase in our production, going from about 1.5 grams per liter of BDO in the wild type strain with the complemented heterologous pathway to almost 3 grams per liter. When we take the same guide RNA cassette and just throw it into another yeast strain, in this case, the Sigma strain, we get almost that same fold improvement, but we get a very different y-axis here, right? So here the baseline is sitting above 5 grams per liter, and we're getting above 10 grams per liter production of BDO. Same pathway design, same guide RNA design, just putting it into a slightly different yeast strain. So this really should utility of being able to use these multiplex guide RNA type of approaches to program and prototype cellular systems. So very rapidly, we can explore genetic diversity, we can explore different strains, and create new types of in vivo-based systems. So that kind of laid the basis for the fact that we can engineer cells rapidly. How can we now use that and move into rewired lysates, where we want to move into in vitro-based systems and allow us to harness the overall capacity for doing rapid catalysis there and for making new products very quickly and for prototyping new types of ways for how things work within an in vitro metabolic system. So really our vision for that is to create a series of different types of strains. I'm going to showcase exclusively today engineered yeast, but we are doing this as well for bacterial systems. Engineer these yeasts, grow these up, lyse them, and take the cell extract, and be able to enable rapid biosynthesis of molecules. And I have 3 case studies, some of which you should already recognize here in terms of the BDO and Glycerol. I'll also talk a little bit about ataconic acid as well. But then this is kind of the first generation aspect. Second generation is, can we actually get these rewired cells, add together into this extract the pathways we want to rapidly and on-demand biosynthesize molecules, as well as understand the underlying pathways. So I should mention that this is work that has been enabled over the past couple of years through the JGI and DOE ETOP program. And this is also work that's in collaboration with the Jewett Lab at Northwestern, especially Blake Rasor, who is a graduate student in the Jewett Lab, and obviously Mike as co-PI on this particular project. So what we wanted to do is kind of first off see, does cell-free metabolism work? Can we metabolically engineer a cell to create a better extract? So to do that, we kind of took that motivation back from that 2,3-butanediol experiment that I showed a few minutes ago, and said, can we take this cell, can we lyse it, put into an extract, and by adding additional substrates and cofactors, can we activate that metabolism? So first off, we wanted to see, can we activate some kind of heterologous pathway to start with? So to do that, we complemented just that BDO pathway itself, so just the heterologous pathway, none of the CRISPR rewiring there yet, and found indeed this is starting to work. When we have new pathways in that cell-free lysates, that free metabolism, the in vitro-based system, has that same type of metabolic capacity. So we can see that when we've complemented that cell produces BDO, and it produces a little bit less ethanol as a byproduct because it's just shifting that carbon over there. So with that fact, we saw that we can indeed activate metabolism in these cell-free systems. So now the next question, does a rewired cell lead to a rewired lysate that would also be better? So we took that rewired BDO strain, and this is through the CRISPR-based modifications that I just talked about a little bit ago that we can rapidly prototype in cells, and ran through this experiment. We looked at wild type, the BDO complemented pathway, and the rewired cell with the BDO pathway, created cell-free lysates for each of these conditions, and looked and see what we had. And as kind of expected, we did see this bump in BDO in our rewired lysate, and we also saw this ever decreasing amount of ethanol. So going from our wild type to our heterologous pathway complemented to our rewired cell. So really what we started to see is that rewiring an in vivo-based system to then make the in vitro-based system seems to translate over well. So how does that expand? Well, we can expand to various different strains, right? So just like I showed that we can rapidly rewire strains, including a BY4741, SNPK, Sigma strain, we can introduce those CRISPR rewirings and that pathway into each of these strains, grow those up, create those into a cell-free system or lysate, and then activate the metabolism there. And in each of the cases, we saw that the rewired cell had an improvement over the background strain. And we also saw that there is this decrease in ethanol as we moved from a wild-type lysate background to a rewired lysate background. The other thing that's beneficial, as I talked about in the introduction, is that we can actually rapidly prototype in ways that we can't do in vivo when we have an in vitro system. So we took this BDO lysate, and we can very quickly change that composition of proteins that's in there. So by changing the extract concentration, we can actually see that something different happens. At lower concentrations, we tend to have a lot of our byproduct, this acetylene that's in that BDO pathway, being formed. As we have more and more protein content, we begin to see that that decreases. But the flip side is that we begin to see that the ethanol concentration in our reaction increases as we have more extract concentration. So there's this balance between having an incomplete reaction and too much ethanol that's produced. So we found that somewhere around the 6 milligram per milliliter concentration of extract seemed to be best. So we can change protein composition pretty quickly in an in vitro system. Can't do that in vivo. Next thing we can do is change the pH. We don't have to worry about cells growing and cells surviving, so we're able to very quickly change the pH of the reaction. And we found that pH of around 8 actually seemed to be most optimal, meaning that we started increasing our BDO content pretty rapidly to around 8, and that seemed to be this trade-off with ethanol, whereas at lower pHs, we tend to make much more ethanol than BDO. As we reached 8, that kind of seemed to be the optimal point. As we moved above that, pretty much metabolism in that cell-free system sort of stopped in terms of being outside of that optimal range for those enzymes. But again, we have the ability to change that since it's an in vitro system. We can change all the cofactors very easily by just mixing and matching and adding them. And we ended up finding that there is a composition of NAD and ATP that seemed to be optimal for this reaction to occur. And then we can also change the initial amount of substrate. And just like when we feed excess sugars to cells, when we fed excess sugars to this in vitro system, we tended to see that there was an overflow of ethanol and not as much of our product. It kind of plateaued, as you can see, at the higher concentrations of glucose. So we found that anything less than about 100 was more favorable for BDO production. So when we do that, we can actually scale this up to and look at a time scale and a time lapse for this particular reaction. We very rapidly utilize our glucose and produce our BDO in a very short period of time. We can do fed batch type of operation for this in vitro system to ever increase that total concentration. And at the end of the day, our in vitro system works much quicker than an in vivo system, produces much higher productivity. So going again to that the presentation from just a bit ago for the keynote for this morning, looking at this idea of creating high productive cells, well, we can do that through these cell-free based systems. So we wanted to see if we can expand to other products. So we took alternative pathways. Talked about glycerol before. We can very rapidly prototype glycerol in vivo by using our CRISPR-based systems. So we had a wild type cell and we had a rewired cell. And we can actually then take that, make lysates from those. And as expected, our strain was able to, our lysate rather from the enhanced strain was able to produce substantially more glycerol and less ethanol than our starting point. And we're able to optimize that in a time course as well, where we're able to get significant amounts of glycerol. That was a high flux, more native pathway that we can rewire. We also looked at the same type of thing for itaconic acid. I hadn't talked about that in the presentation yet, but really we were able to rewire this through a series of different mutations that we had published on a couple of years ago and be able to go from a base strain and almost double the amount of itaconic acid that can be produced in that in vitro lysate from our rewired cell. So just kind of digging in here, once again, we can go through the rapid prototyping. We can actually go through and change cofactors, temperatures, the concentration of sugars. And when we do that, we're able to get very high amounts of glycerol, as I mentioned, and in very short periods of time. So here we're able to get much shorter time for the reaction compared to in vivo. In vitro titers were much higher than that of in vivo, and likewise, almost an order of magnitude higher productivity that we were able to get by using these cell-free systems for the production of glycerol. Same thing holds for itaconic acid. A couple of different cofactors we had to explore just because of the complexity of this pathway, but nevertheless, we're able to find combinations that really serve to increase that overall production. And in doing so, we were able to get almost that order of magnitude increase in terms of the productivity associated with that in vitro system, especially because it can happen so rapidly. So with those examples, hopefully you've been able to kind of provide some motivation here that a cell-based system, cell-free systems, we can integrate these together through the tools of synthetic biology and metabolic engineering and the technologies, especially with JGI in the DNA synthesis core. And we see that CRISPR tools, these are scalable technologies that can be multiplexed and easily reused and moved between different cells. And that allows us to create various different platform strains that we can be able to rapidly test and also rapidly transition over into cell-free systems. Likewise, these rewired lysates enable really enhanced biosynthesis, especially in terms of the productivities and the ability to be able to modulate those environments in ways that you couldn't do within a cell. And at the end of the day, this coupling of this in vivo and in vitro system really, we feel, opens up great opportunities, not just for synthetic biology prototyping and gene discovery and functional genomics, but also for a new area of cell-free based biomanufacturing of small molecules. And as I kind of alluded to, motivations moving forward, we're working on creating a variety of these different cell-free frameworks to be able to rapidly go through this design, build, test cycle in ways that we can really enhance the technologies that are going on and offered at JGI, but then also to look at new ways for the rapid prototyping of pathways of interest for the various different systems. So with that, I really like to thank the contributors to this work, in particular, the labs, both my lab as well as Mike Jewett's lab, and our funding for this work, especially JGI and their great support. And happy to be able to have this opportunity to present today. So thanks again for the invitation, and I'll open it up for any questions. I'll stop sharing so I can see the screen and have discussions from there. So thank you very much.
Well, as always, Hal, that was absolutely fantastic. We have a couple of questions that came into the Q&A box. Firstly, how did you decouple the potential burden imposed by expressing dCas9 from other intended effects? Yep, sorry, that's the question.
Oh, okay. Yeah, so it's a great question there. So what we were able to do is really go through this kind of funneling, as I kind of showed on there, in terms of the design selection. So we were able to go through and find guide RNAs that were really not going to hit anywhere else in terms of off-target effects, that didn't interfere with transcription factors, that really didn't provide much of a burden when we express that activator or repressor. And I kind of talked a little bit about kind of the streamlining that we were doing in terms of things. So the streamlining in terms of how we express our guide RNAs, the streamlining in terms of how we express our dCas9-based system, that's really kind of led to a system that has minimal burden overall. But at the end of the day, it is both a rewiring tool, but also a discovery tool. So it is that trade-off at the end of the day of being able to what you want is your final strain, whether you want it to be a final strain that is CRISPR rewired or a final strain that is genetically encoded based off the information you get from that CRISPR system.
Excellent. So there was a lot of discussion about scaling the engineering, and with multiplex CRISPR, how you can very rapidly explore huge amounts of functional space. But that is, to a degree, contingent upon the scaling of the downstream assay. So can you comment on where the compromises would be if the downstream assay did not scale to the same degree as the engineering?
I'd say that we need better downstream analysis tools. And I kind of alluded to that very quickly. So we have a paper that should be published sometime this week where we've actually started to expand into microfluidic and droplet-based sorting. And that has allowed us to get to 10 to the 7th type size libraries very rapidly in the course of a half a day for analysis of secreted metabolites. And when we're talking about some of these CRISPR-sized libraries, we're well within that range to be able to kind of sort through those types of library sizes in a very quick, facile manner. So I think the technologies are getting there. I think we've demonstrated even before that, that's for example, the production of beta-xanthins, we're able to screen through the libraries that were created at JGI in a very rapid fashion by just looking at microtiter plates. It's not exhaustive. It's a little bit more laborious, obviously, on there. But there are different types of means to be able to kind of look at these overall types of libraries.
Great. Okay. Is separating out the products from a cell-free system harder than it would be from an in vivo system?
Yeah, so it's a good question. For the molecules that we've looked at, not much of a difference in that regard. For certainly for various different types of removal of proteins, this tends to be easier because you kind of can enrich really that cell-free system and run a dialysis first and kind of minimize the amount of contaminants over there. So the molecule really is kind of the driving force of that downstream separation. And it's really the chemistry that you're creating from there. But you are creating a very enriched environment in terms of that concentration. So it is the molecule that's really is the biggest thing that's out there in that milieu.
Okay, great. And just one final question here. Can this in vivo approach be used to activate or enhance lipase production in yeast cells?
Yeah, so I think that's it's an interesting question, and I think the use of kind of in vitro based systems as screening platforms, I think, is something that's that would be interesting on there, and people have actually looked at things like micro droplet systems or micro well separations, and kind of using those cell-free based systems as ways to kind of both look for increased protein production, look for increased protein activity. So I think we're going to start to see a new set of tools for high throughput screening and selection. So certainly, I think that lipase sensitivity assays can be done in vitro. Would be a pretty interesting type of approach.
All right, fantastic. That covers all of the questions that came up in the Q&A box. So I'm just going to round this off by thanking you again, Hal. Absolutely fantastic. And take care, everybody.